Cells are incredibly small, but they function like highly organized chemical factories. Inside each cell, thousands of chemical reactions occur simultaneously. These reactions allow cells to maintain internal balance, manufacture cellular components, and replicate when necessary. All of this activity requires energy. As cells perform work, they continuously transform energy from one form into another to sustain life.
Energy is defined as the capacity to cause change or perform work. There are two major types of energy. First, kinetic energy, the energy of motion. Second, potential energy, energy stored as a result of an object's position or structure. In biological systems, both forms are essential, but much of life depends on stored potential energy that can be released when needed. Heat, or thermal energy, is a form of kinetic energy associated with the movement of atoms and molecules. Light is another form of kinetic energy and can be harnessed by plants to power photosynthesis. Chemical energy, however, is the most important form of energy for living organisms. Chemical energy is stored in molecular bonds and can be released during chemical reactions. This stored energy powers the work of the cell. Thermodynamics is the study of energy transformations in matter. When studying energy changes, scientists refer to the system, the matter under investigation, and the surroundings, which include everything outside the system. Understanding how energy moves between the system and its surroundings helps explain biological processes. Two laws govern energy transformations in organisms. The first law of thermodynamics states that energy cannot be created or destroyed, it can only be transformed. The second law of thermodynamics states that energy conversions increase the disorder of the universe. This disorder is called entropy. Entropy is a measure of randomness or disorder, and it always increases in natural processes. Chemical reactions either release energy or require energy input. Exergonic reactions release energy. Endergonic reactions require an input of energy and store energy in chemical bonds. In living systems, exergonic reactions often power endergonic reactions. This coupling allows cells to build complex molecules and perform essential biological work. Exergonic reactions release energy. In these reactions, energy stored in the covalent bonds of reactants is released as the products are formed. For example, burning wood releases the energy stored in glucose as heat and light. Cellular respiration is also an exergonic process. It occurs in many controlled steps, releasing energy gradually rather than all at once. Importantly, some of this released energy is captured and used to produce ATP. This diagram illustrates an exergonic reaction. The reactants begin with a higher level of potential energy. As the reaction proceeds, energy is released. The products end with lower potential energy than the reactants. The difference between the energy levels represents the amount of energy released during the reaction. In biological systems, cells capture part of this released energy to perform work. An endergonic reaction requires an input of energy. These reactions yield products that are rich in potential energy. Endergonic reactions begin with reactant molecules that contain relatively little potential energy. They end with products that store more chemical energy than the starting materials. Because energy must be added, endergonic reactions do not occur spontaneously. This diagram represents an endergonic reaction. Here, the reactants start with lower potential energy. Energy must be supplied for the reaction to proceed. The products end with higher potential energy than the reactants. The upward difference in energy represents the amount of energy required. In living cells, energy from exergonic reactions is often used to drive these energy-requiring processes. Photosynthesis is a classic example of an endergonic process. Energy-poor reactants, carbon dioxide and water, are used. Energy from sunlight is absorbed. This energy is used to produce energy-rich sugar molecules. In this way, light energy is converted into stored chemical energy within glucose. A living organism carries out thousands of endergonic and exergonic chemical reactions every moment. The total of all these reactions is called metabolism. Metabolism includes every chemical process that builds, modifies, or breaks down molecules inside the cell. A metabolic pathway is a series of chemical reactions that either builds a complex molecule or breaks a complex molecule into simpler compounds. These pathways are carefully regulated to maintain cellular balance. Energy coupling allows cells to drive essential reactions. It uses the energy released from exergonic reactions to power endergonic reactions. In most cells, this coupling occurs through ATP. ATP acts as an energy intermediary, capturing energy from energy-releasing reactions and transferring it to energy-requiring processes. Without energy coupling, many critical cellular reactions would not proceed. ATP, adenosine triphosphate, powers nearly all forms of cellular work. ATP consists of three components: the nitrogenous base adenine, the 5-carbon sugar ribose, and 3 phosphate groups. The bonds between these phosphate groups store potential energy. When ATP is hydrolyzed, energy is released and made available for cellular activities. Hydrolysis of ATP releases energy by transferring its third phosphate group to another molecule. This process is called phosphorylation. When ATP loses a phosphate group, it becomes ADP, adenosine diphosphate. Most cellular work depends on ATP energizing other molecules by phosphorylating them. This energy transfer makes otherwise unfavorable reactions possible. This diagram shows ATP composed of adenine, ribose, and 3 phosphate groups. When water is added, in a hydrolysis reaction, the terminal phosphate group is removed. ATP becomes ADP, and energy is released. This released energy is then used to power cellular processes. The cycling between ATP and ADP is continuous in living cells. There are 3 main types of cellular work. First, chemical work, such as building macromolecules. Second, mechanical work, such as muscle contraction or movement of cellular components. Third, transport work, such as moving substances across membranes against a concentration gradient. ATP drives all 3 of these types of work. Through energy coupling, ATP ensures that cells can grow, move, maintain organization, and survive. This diagram illustrates how ATP drives 3 types of cellular work. First, chemical work. ATP provides energy to build molecules by phosphorylating reactants. Second, mechanical work. ATP powers motor proteins that move structures such as protein filaments. Third, transport work. ATP fuels membrane proteins that move solutes across membranes against concentration gradients. In each case, ATP is hydrolyzed to ADP and inorganic phosphate, releasing energy to perform work. ATP is a renewable source of energy for the cell. In the ATP cycle, energy released from an exergonic reaction, such as the breakdown of glucose, is used in an endergonic reaction to regenerate ATP from ADP and phosphate. This cycle allows energy to continuously flow through the cell. ATP is constantly being broken down and rebuilt. This diagram shows the ATP cycle. Energy from exergonic reactions is used to add a phosphate group to ADP, forming ATP. When ATP is hydrolyzed, energy is released and used to power endergonic reactions. The cycle repeats continuously, linking energy-releasing and energy-requiring processes. Enzymes speed up chemical reactions by lowering energy barriers. Although biological molecules contain stored potential energy, that energy is not released spontaneously. An energy barrier must first be overcome before a reaction can begin. This required energy is called activation energy, often abbreviated as E sub A. By lowering activation energy, enzymes allow reactions to proceed more quickly and efficiently. Here, we see the concept of activation energy illustrated graphically. Activation energy is the amount of energy required to move reactants to an unstable transition state. Without enzymes, this energy barrier is high. With enzymes, the barrier is significantly reduced. This does not change the overall energy released, it simply makes the reaction occur faster. Each enzyme functions best under specific conditions. Temperature affects molecular motion. An enzyme's optimal temperature produces the highest rate of contact between reactants and the enzyme's active site. Most human enzymes function best between 35 and 40 degrees Celsius. pH also influences enzyme activity. For most enzymes in the human body, the optimal pH is near neutral. Many enzymes require non-protein helpers called cofactors. Cofactors bind to the active site and assist in catalysis. Some cofactors are inorganic, such as zinc, iron, or copper. If the cofactor is an organic molecule, often derived from vitamins, it is called a coenzyme. These helpers are essential for proper enzyme function. Enzyme inhibitors regulate enzyme activity within cells. A competitive inhibitor resembles the substrate and competes for binding at the enzyme's active site. When the inhibitor occupies the active site, the substrate cannot bind. This reduces the enzyme's productivity. However, increasing substrate concentration can sometimes overcome competitive inhibition. Non-competitive inhibitors bind to the enzyme at a location other than the active site. This binding changes the shape of the enzyme. As a result, the active site is altered and the substrate cannot bind effectively. Unlike competitive inhibition, increasing substrate concentration does not overcome non-competitive inhibition. Enzyme inhibitors play an important role in regulating cell metabolism. In some pathways, the final product acts as an inhibitor of an earlier enzyme in the pathway. This process is called feedback inhibition. When enough product accumulates, it shuts down further production. This prevents waste and maintains metabolic balance.